Good evening, everybody, and welcome to our Keynote Session 2, which is a very exciting session. My name is Terry Magnuson from UNC Chapel Hill, and I am the immediate past president of the GSA. And so we have two keynote speakers tonight and one awardee for the Morgan Medal. So we're going to start with our first keynote speaker, and it's Elaine Fuchs from the Rockefeller University. And I've known Elaine for a long time. Her work is focused on adult stem cells that reside in all tissues and where they replenish dying cells and repair wounds. Using mammalian skin as a model, Elaine has been studying the remarkable properties of tissue stem cells and how they know which tasks to perform and when. She explores how stem cells sense, communicates with other cells in their environment, and has been dissecting how communication networks malfunction in inflammation, aging, and cancers. Elaine's talk is entitled Stem Cells in Silence, Action, and Cancer. So welcome, Elaine.
Great. Okay, hopefully everyone can see this and see me. And let me just start by thanking the TAGC for holding this conference in the face of the unbelievable upheaval that our world has faced over the last several months. And I think it's just a wonderful venue to really bring us all together. And I always think that I like to start my meeting by showing you pictures of a recent trip that I took. And I think this time it's particularly poignant because it happened right before the pandemic broke. We were in the Galapagos Islands, and it's a beautiful position to remind you that nature clearly has had a lot more fun and fancy in creating body surfaces than she has in creating any of the ugly organs that most of you work on. And so for me, I've always been fascinated by the body surface and by the diversity, tremendous diversity that it undergoes. So we don't dig very much deeper into the skin. We go to the surface epithelium. The skin epithelium is one of the body's primary environmental interfaces, and it's subjected to all sorts of different stresses, from oncogenic stress, ultraviolet radiation, wounds, mechanical stress, pathogens. And I just point you to temperature and pH extremes. We don't often think that our body surface is actually pH 5, where most of our cells are growing at pH 7.4. And recently, very recently, the last couple of weeks, we've published a paper where we've learned that these temperature and pH extremes basically prime cells with or proteins with unstructured domains to undergo liquid-liquid phase transitions that turn out to drive the process of epidermal differentiation. A really fascinating story, and just one example of how stem cells of the skin have to be poised to be able to sense these changes that it faces and to rapidly adjust, and in this case, exploit them. And so over the years, we've isolated and characterized the stem cells that exist within the skin epithelium. We've shown that they reside at the interface between the epithelium and the mesenchyme. And so epidermal-dermal interactions play an important role in driving what these stem cells are going to do. But in addition, what we find is that the stem cells, whether they're in the epidermis, the sebaceous gland, or the hair follicle, are also receiving different types of stimuli from other cell types within the skin. They each have their own specific niches, and it turns out that their molecular properties, as well as their tasks, are they going to make epidermis, sebaceous glands, or regenerate the hair follicle and grow hair, are determined by their interactions with their local niches. And I point you to another paper, in this case published at the end of this past year, where we're still uncovering the complexity of stem cell interactions with their local environment, and in this case, demonstrating that the stem cells of the hair follicle are all interconnected across the tissue by a network of lymphatic capillaries, and that coordinates stem cell behavior so that the stem cell niches can basically react and respond to the particular demands. So we've been interested in understanding how the stem cells perceive and respond to their microenvironment. And in work done by Renee Adam and Hansel Young in my laboratory, we've dissected mechanisms by which the stem cells receive growth factors from, or stimulatory factors from their environment, and they respond at the level of chromatin dynamics. And so these two graduate students mapped out the binding of all the different transcription factors that are expressed by the hair follicle stem cells, along with various different epigenetic marks, histone modifications, and mediator that brings the enhancer together with the promoter. And what we found is that within these large open chromatin domains called superenhancers, there are the binding platforms for the binding of all these different transcription factors. And it turns out that it's only about 5% of all of the genes that are expressed by the hair follicle stem cells that are regulated by superenhancers, but it's a very important group of genes. These are the genes that are sensing the microenvironment that the stem cells are in. And so the way in which they do that is that they not only bind all of the different transcription factors, but they also bind the DNA effectors for the signals that these stem cells are exposed to. In the case of a quiescent stem cell sitting in a hair follicle stem cell niche, there is a high BMP level, and that BMP level has to be downregulated. Wnt signaling has to be upregulated in order to mobilize these stem cells from a quiescent state where they're not making tissue to one where they are making tissue. And it turns out that the DNA effectors for BMPs and Wnts are also packaged within these short regions of chromatin. So effectively that makes these regions, which we call epicenters, molecular zip codes for this is who I am, a hair follicle stem cell, and this is where I am in a quiescent stem cell niche. So what happens then when we take these stem cells out of their environment? We can put them into culture. We know they're still stem cells because we can graft these stem cells after culture onto a hairless mouse and regenerate hair. So they're still stem cells, but while they're in culture, those 5% of genes that are regulated by superenhancers change dramatically. Most of them are turned off, and a new group of superenhancer regulated transcripts are turned on. They turn out to be regulated by transcription factors that are induced in a wound state. We're taking these cells from a quiescent environment, putting them into serum-rich, growth factor-rich media where the stem cells are dividing, and it turns out that they now act as if they're in a wound-induced environment, which we've gone on to demonstrate by marking the stem cells and monitoring them as they repair wounds. And so how can we also then take the data, the high throughput chromatin landscaping that we've been obtaining, and be able to test the physiological relevance of our findings? The way in which we do that is to cut out these regulatory elements, these superenhancer regulatory elements, use them to drive GFP. And we package this gene then into a lentivirus, and now we can just inject the lentivirus into the amniotic sac of a 9.5-day mouse embryo that has at that time just one layer of skin. It's a surface layer of progenitor cells, and those progenitor cells are going to develop into all of the skin epithelia. If we do that then and do it in living embryos and now allow those embryos to grow up into adult mice, now we find that the EGFP reporter is expressed only in the hair follicle stem cells and only in the quiescent niche. So if we take these cells and remove them and put them into culture, they shut off the transgene. If we graft the cells back on and they reform the stem cell niche, they turn it back on again. And now we can take one of those in vitro epicenters. And if we make a mouse driving EGFP, in the unwounded state, there's no activity. But when we now wound the skin of these animals, we get EGFP. When the wound is repaired, the EGFP shuts off again. So these data really tell us and give us a way in which we can test the physiological relevance of these high throughput data that we're discovering. So what happens then in cancer? Cancer as a wound that never heals. I've already shown you that in a wound, there are unique chromatin regulatory elements that are opened up at a wound site. And it turns out that if you then look at what happens in cancer, you find that there's tremendous parallels between the wound-induced state and the tumor-induced state, but now we see new chromatin peaks that open up. Those chromatin peaks are invariably regulated by a transcription factor, ETS2, and one of the things that we've learned is that in a transient wound, there is a low level of RAS-MAP kinase pathway that is upregulated. Once the wound is repaired, it's turned off again. And during that period of time, ETS2 gets phosphorylated by MAP kinase. But in a cancer, particularly squamous cell carcinomas, now there's typically mutations in the RAS genes. And now what we find is that RAS-MAP kinase is sky high, as is ETS2, and that then leads to the opening of chromatin. So again, we can test the physiological relevance of our findings by taking one of those squamous cell carcinoma specific regulatory elements. In the unwounded state, no activity. In the wounded state, no activity. And in squamous cell carcinoma or malignant progression, now the transgene turns on. So this provides us not only with demonstration of the relevance of our findings, but it also provides us with a number of new tools with which we can now probe more deeply the states of these cells. So I want to now switch and focus the rest of my talk on the environmental impact and changes in malignancy that go beyond simply genetics. So this is a genetics meeting, and clearly oncogenes and tumor suppressors override some of the effects of the environmental impact on malignancy, but they don't completely get rid of it. And so here we focus again on squamous cell carcinomas, which are the second most common, sixth most deadly cancer worldwide. They affect not only the skin, but also a variety of other epithelial tissues. And I just point out that for patients that are provided with immune suppressive drugs, they dramatically increase the risk of squamous cell carcinoma, making it more deadly than that of, or as deadly. And now I'm not finding that this is working, as deadly as, okay. I'm going to restart the program because my computer has somehow frozen. I think there's, okay. Okay, so let me quickly go on. To say what we've done then is to isolate and characterize the tumor-initiating cells of squamous cell carcinomas. We know that they can be derived from hair follicle stem cells as they acquire mutations in Harvey-RAS. And we also know by serial dilution and analysis of the cells within squamous cell carcinomas that the tumor-initiating cells reside right at the interface between the tumor and the stroma. very similar to what you saw for normal stem cells, but now the stroma is very different. Cancer-activated fibroblasts, immune cells, a whole variety of differences that basically render the microenvironment of the stem cells very different from that of the normal stem cells. And in addition, what we know is that there is angiogenesis going on at tumors, and wherever a blood vessel comes up near these tumor-initiating cells, it brings with it a milieu of rich TGF-beta coming from a number of the immune cells. And what that does is change the local microenvironment, not of all the stem cells, but of those that are near a blood vessel. It turns out that TGF-beta signaling now impacts the behavior of the tumor-initiating stem cells that are nearby. And the way in which we began to monitor and track the behavior of these stem cells was to engineer a mouse where wherever a blood vessel came up next to the tumor, such as the area over here, then the tumor-initiating cells fluoresced cherry in response to the TGF-beta signal. Over here, there's no TGF-beta signal, and the cells are basically not near a blood vessel. We also worked out a method where we could turn these fluorescent red cells to green and lineage trace them. If we isolate the cells, these TGF-beta sensing cells have divide more slowly than their normal wild type counterparts. But that means then that most of the tumor cells over here are growing the tumor very quickly. The tumor cells over here that are receiving a blood vessel TGF-beta signal are growing more slowly. But when we lineage trace them, we see that it is these cells receiving a TGF-beta signal that are breaking down the basement membrane and invading. And remember, the blood vessel is right here. So these cells are growing the tumor very rapidly. These cells are invading. That's all happening in one genetic background in the same tumor due to changes in the microenvironment. Does it matter? We can give these cells a chemotherapeutic drug, in this case cisplatin, which is the drug of choice for head and neck cancers. And we see by caspase-3 in blue that very rapidly most of the tumor dies. But if we look at the TGF-beta responding cells, we see that they are not undergoing apoptosis. Are they actually important in the process of tumor regrowth? What we find is that if we wash away cisplatin, activate the lineage tracing marker, and now look for tumor regrowth, and when the tumor regrows, what we look for is the tumor green? And it is, indicating that it's coming from the TGF-beta responding tumor-initiating stem cells. So what about these cells with regards to immunotherapy? Here in the work of Phoenix Miao in my laboratory, we took advantage of an existing mouse where we could isolate activate T cells that are specifically targeted to attack a cell that has an OVA antigen on its surface. We then engineered a mouse which has two tetracycline-inducible regulatory drivers, one to drive Harvey-RAS mutant, and the other is to drive OVA. We then introduce a lentivirus at low titer expressing the tetracycline activator. So now, after the animal grows up, we can start administering doxy. That induces the tumors in a very small number on the body surface, but each one of those tumors expresses OVA. And if we now add these cytotoxic T cells to the tail vein of these mice, we see that the cytotoxic T cells attack the tumor in response to their expression of OVA. So we look at what happens. Within a few days, 90% of the tumor cells are attacked and cleared, but we could never get rid of the final 10% of the cells. So if we take a look at what are those cells, we showed that additional doses of cytotoxic T cells don't help, and eventually the tumor grows back. What are the cells? The cells turn out to be the same TGF-beta responsive initiating cells. In a naive tumor, they're less than 2% of the total cells within the tumor. After 1 week of immunotherapy or CAR T therapy, what we find is that they're now about a quarter of the remaining cells. And then we can do our lineage tracing experiment, in this case using a red marker rather than a green one. But when the tumor grows back, it is coming from these TGF-beta responsive cells, which are now refractory to, in this case, immunotherapy. So what's a molecular basis for this? It turns out to be 2 different mechanisms. The first mechanism, with regards to chemotherapy, is that one of the targets of TGF-beta is P21, which can interact with KEAP1, an inhibitor of NRF2, which activates the glutathione pathway. So when NRF2 then gets activated, the genes and the pathway get very highly upregulated, and that's what clears out nasty things like cisplatin from the cell. If we look at human head and neck cancers, what we find is a small cohort of them have highly upregulated glutathione pathway genes, and those patients have the poorest prognosis. So what about TGF-beta signaling and immune resistance? It turns out to be a different mechanism. In this case, there is a family member of the PD-L1 family of ligands, in this case CD80, that turns out to be expressed by the tumor-initiating stem cells at the right amounts, so that at low levels, CD80 on the surface of these cells interacts exclusively with CTLA4, and that dampens down and renders the activated T cells incompetent to be able to attack. And so in this case, we also take a look at human databases, and what we find is that a small cohort of patients with squamous cell carcinomas, the carcinomas have CD80 on the surface. That also leads to poor prognosis. So both of these mechanisms turn out to be important, and they're environmental mechanisms for changing the program of the cells in order to be able to drive tumor resistance. So now let me finish my talk by going to yet another aspect that doesn't deal with genetics, and that is, what about translational control? In the work of Ataman Sendwell, a postdoctoral fellow in my laboratory who's since gone on to EZH in Zurich, what we found is that the cancer stem cells turn out to have dampened down protein translation relative to wild-type stem cells and relative to other cells of the tissue. So what happens, however, is that in the face of global protein translation repression, what we find is still a number of different messenger RNAs that are efficiently translated. They turn out to be efficiently translated by virtue of the fact that they have unusual 5' open reading frames in their 5' UTRs. What that does is that leads to effective translation. So how does it do it? It turns out that in oncogenically stressed cells, they activate the integrated stress response, and that leads to the phosphorylation and inactivation of the normal canonical translational effector. Instead, what happens is that EIF2A, a promiscuous non-canonical effector, becomes essential. And if we knock out EIF2A in the squamous cell carcinomas, we can show that the tumors now are unable to grow in the absence of this secondary translational effector. So finally, EIF2A is amplified in cancers, and we also find that in about actually 20% or more of squamous cell carcinomas, that the EIF2A locus is amplified. This locus contains other oncogenes, but if we take a look specifically at the levels of EIF2 messenger RNA in these cancers, what we find is that those that had the highest levels of eIF2A messenger RNA specifically have the poorest prognosis. Again, pointing to the realization that the work that we're uncovering in terms of understanding how tumor-initiating stem cells avoid various different stresses, environmental stresses that they're faced with, is basically giving us a clue as to the basis of cancer resistance. So finally, I'm just going to finish by dealing with one last issue, and that is, as we were beginning to realize that oncogenically stressed cells dampen down protein synthesis, we began to wonder whether they also adjust their metabolism in order to maintain their oncogenic state. And that turns out to be the case. And in this case, Sanji Baksh, a talented MD PhD student in my laboratory, in collaboration with Lydia Finley, running her own laboratory at Sloan Kettering, teamed up to sort this out. And what we learned is that simply by controlling the animal's diet, we could dramatically dampen the tumor burden on these animals. So how do we do it? It turns out that oncogenically stressed tumor cells become reliant upon exogenous serine. They could make their own serine. It's not an essential amino acid, but they don't. Why don't they? It turns out that if they're forced to do so, then they end up producing higher levels of a key metabolite, alpha-ketoglutarate. And alpha-ketoglutarate is an essential cofactor for a number of histone demethylases, as well as DNA demethylases, as well as HIP-1 alpha. It turns out in this particular case that by dampening down serine glycine in the media or in the tumor, what happens is that now those demethylases are activated, and specifically histone K23 trimethylation is demethylated. And when that happens, now the tumor cells undergo precocious differentiation. So this is the mechanism then. Alpha-ketoglutarate is utilized in the, or is produced in the serine biosynthesis pathway. The tumor cells avoid that from happening and basically are able to survive. And if they don't, they undergo differentiation, and that leads to the loss of the tumor. So cancer then is really a tango, not only of genetic, but also environmental impacts. The prospects for therapy, I think, are appreciable. We know that RAS-MAP kinase has been a difficult druggable target, but there certainly have been advances made in this area. We know that ETS2 phosphorylation occurs as a consequence of elevated levels of RAS-MAP kinase, and it might be a druggable target. We know that eIF2A, a non-canonical initiator, is not necessary for the normal stem cells, so maybe by inhibiting it, we could prevent the cancer stem cells from growing, but still allow no effect to the normal stem cell counterparts. With regards to TGF-beta, there it's a bit of a difficulty in terms of thinking how you would use TGF-beta inhibitors in terms of chemotherapy. We know that TGF-beta is a tumor suppressor, and we know that the stem cells, if they're seeing a TGF-beta signal, will dampen down the rate of division. So if we were to simply start by inhibiting with TGF-beta inhibitors, we would probably end up with very rapidly growing tumor cells that now could acquire additional mutations. They don't do that because of the mechanisms that I've described. So we wouldn't want to start with TGF-beta inhibitors, but we'd want to start with chemotherapy to wipe out the majority of tumor-initiating stem cells, then come in with TGF-beta inhibitors to mobilize those stem cells that remain, and then finally come back again with chemo or immunotherapy. And I talked about the potential of CD80 as a new target for immunotherapy. And my favorite one is shifting diet, as it's become increasingly clear that this might be an avenue for future treatments of cancer. So my work would not be possible were it not for the work of a number of really talented people in my laboratory. The ones that are currently in the lab, Phoenix Miao did the work on the immunotherapy, is about to start his own laboratory at the University of Chicago. Shiri Gurkoen in my laboratory did the work on the lymphatic capillaries and is a Human Frontiers Fellow in my laboratory. Felipe Garcia Quiroz is the person who did the interesting work on the liquid-liquid phase transitions in exploiting the environment of the epidermis in order to drive differentiation and produce the skin barrier. Hansel Yang did the chromatin high throughput analysis that I've described, and Sanji Baksh did the work on metabolism. And I finally wanted to highlight Rachel Neek, who is an MD PhD postdoctoral fellow in my laboratory, currently on the lines fighting COVID virus at Sloan Kettering in New York. So with that in mind, I thank the organizers for the opportunity to present this work to you today.
Okay, thank you, Elaine, for really a very stimulating and exciting talk that raises even more questions to keep going. One simple question that someone's asking, we're not supposed to have questions in this format, but it's what kind of diet would be low in serine?
Well, for us, treating the mice, we can easily dampen down serine. You have to dampen down glycine as well, because otherwise the glycine biosynthetic pathway or the glycine production will basically can go the reverse direction and generate serine. That happens in the liver particularly. So you need to be able to reduce both serine and glycine. Obviously, you can't get rid of it entirely. That would be deleterious to the animal. But by just dampening it down, you can actually reduce the levels of alpha-ketoglutarate enough to be able to restore the histone marks that are otherwise allowing to be able to activate enough of the demethylase in order to be able to get rid of those histone marks that would otherwise prompt and fuel the stemness. Glutamine is another source that basically by reducing it can reduce the levels of, can activate the levels of alpha-ketoglutarate or reduce the levels of alpha-ketoglutarate, and you can adjust it in that way.
Okay. Well, thank you, Elaine, and thank you for taking the time and presenting the work that we have quite an audience that are listening right now. So it's been good to see you, and we're going to move on to our next event. And I'm going to share my screen because I'm very excited to be able to present the 2020 Thomas Hunt Morgan Medal to David Botstein of Calico Life Sciences and Princeton University. The Thomas Hunt Morgan Medal is awarded to individual GSA members for lifetime achievement in the field of genetics, recognizing the full body of work of exceptional geneticists. So we could ask the question if, we already awarded the 2020 Morgan Medal yesterday to Jerry Fink. And the GSA board decided that both Jerry and David independently merited the medal. And this year we are awarding the Morgan Medal to the two of them. Now, the last time this happened was in 1984 when it went to George Beadle and Alexander Brink. And one time before that in 1981 when it went to Marcus Rhodes and Barbara McClintock. Now, while a graduate student, David co-discovered transposable drug-resistant elements. His subsequent yeast research has spanned genome structure and sequencing, the cytoskeleton, protein secretion, sugar metabolism, topo2, DNA replication, calmodulin, genomic analysis, and genome databases. He's developed widely used approaches for conditional mutations to define genetic pathways and using transposable drug resistance for genetic analysis. In 1980, David and his collaborators outlined how restriction fragment length polymorphisms could be used to construct linkage maps in humans. And this approach became a cornerstone of the Human Genome Project, and it's allowed polymorphisms to be linked with disease phenotypes, leading to the mapping and cloning of 1,700 disease-related genes, some of which include Huntington's and cystic fibrosis. David then worked with Pat Brown at Stanford and others on the development of microarrays to measure gene expression on genome-wide scale. In 2000, they built on this approach to classify human breast tumors and subtypes based on their microarray gene expression profiles. And this resulted in a direct clinical application in managing breast cancer. David's also a very dedicated and innovative educator. At Princeton, for example, he developed the influential integrated science curriculum that combines biology, physics, chemistry, and computer science, and he's been a very highly successful mentor. His former students and postdocs include a large number of luminaries in the field, and they include 3 other winners of the Morgan Medal. So David's legacy to the field has been enormous, lasting, and on behalf of the GSA, please join me in honoring David for his many, many, many contributions to genetics, science, and medicine. And I'm going to turn it over to David for a few remarks. Are you there, David?
Yeah, I'm here.
Okay.
Okay, you're still sharing your screen.
I am. I
At
stopped.
least it's by me. Okay.
Yeah, you're okay.
Okay. So thank you, Terry, and the selection committee for this great honor. For me, it's especially meaningful for a number of reasons. First, the Morgan Medal recognizes the 2 elements of my career I most personally aspire to. One is the development of genetics as a science that not only studies mechanisms of inheritance, but also provides insights and tools for the understanding of biology quite generally. The other is the teaching of genetics, especially in the laboratory, as the premier exemplar of scientific exploration and reasoning. I like to think of my influence, such as it has been, as primarily indirect, as was Thomas Hunt Morgan's. I'm most happy with the successes of my many lab alumni at MIT, Stanford, and Princeton, several of whom have already been recognized with the Morgan Medal themselves. Secondly, I'm honored by sharing the honor with my great friend and colleague, Jerry Fink. We have been colleagues and occasional collaborators and co-teachers for the entirety of my career. And Jerry, like me, has contributed as much through his teaching as his direct research successes. The yeast molecular genetics community, more generally, has been a wonderful group of colleagues that have made my career a pleasure. Third, of course, I'm honored by the award because the previous laureates were my heroes and mentors, whose work and influence made genetics in general, and me in particular, productive and successful. I cannot think of a greater honor than to join this distinguished group. Thank you again for the great honor and recognition, and for all the work that the GSA does to further our science and understanding.
And John's lab focuses on the mechanisms linking systems-level programs of gene expression and protein function to discrete cell biological processes in developing embryos. He combines systems biology and bioinformatics and novel strategies of in vivo imaging with the goal of understanding the etiology of human developmental disorders. John's talk is entitled "A Liquid-like Organelle at the Root of Modal Ciliopathy." So John, it's all yours.
Thanks so much, Terry. I appreciate it. And thanks so much to the GSA for giving me the opportunity to present your research, my research this evening to your audience. A great job putting this conference online under less than ideal circumstances in an amazingly quick turnaround. I want to say a huge congratulations to David. It's humbling really to be here giving this talk after David and Elaine. I also want to give a quick shout out to my laboratory. Thanks so much for all you've done. Handling this shutdown with courage and grace has been an inspiration. You know, these are really trying times, and I'd like to put it out there to the PIs to sort of be aggressively compassionate with your trainees right now. I think there's a real danger of losing like a large part of the next generation of biologists, and we can't afford to have that right now. We need these people. And so I hope people will really take that to heart. Okay, so for the past few years, I've been making the rather controversial claim that all of us are developmental biologists. No matter what you're studying, you're a developmental biologist as well. And the reason I say that is because, you know, if you want to study lung physiology, you really can't do that until the embryo, specifically the endoderm, has constructed a lung for you to study. We can't probe consciousness until we actually understand how the embryo makes a brain. And so when you think about it that way, I like to say that developmental biology really is the, well, it's the inability to advance the slides, apparently. Huh. Let's see. This is disappointing. Or to escape from this, yeah. Okay, there we go. That ultimately, developmental biology is the longitudinal and multi-scale integration of genotype and phenotype. That is, that you have this giant constellation of genetic interactions, protein-protein interactions, metabolomics, et cetera, et cetera. And we need to understand how all of these things work together to build tissue architectures and also individual cellular architectures. But we can't really stop there, because it's not just the architecture that matters, but the actual function. So this cell here is a multi-ciliated cell. And as beautiful as the structure is, unless it can do this, which is to beat 20 times per second for its entire lifetime, always in the same direction, then what it can't do is do this, which is to generate fluid flows. And if it can't generate fluid flows in the tissue, then this entire tissue architecture is really rather meaningless. And so today I'm going to talk about my lab's recent efforts to sort of use the multi-ciliated cell as a microcosm of developmental and cell biology, and really take it from the genomics and systems biology level straight down to the function of the cell. Now, multiciliated cells are an important cell population. You've got some in your brain ventricles where they move cerebrospinal fluid. You've got some in your airway where they're central for mucus clearance. About half the population has them in their fallopian tubules. The other half has them in their seminiferous tubules, where they move eggs and sperm during their maturation. And in case you're wondering, yes, a paper came out just recently, COVID-19, the SARS-CoV-2 virus infects these cells. That's not something that I'm going to be focusing on today, but rather I do want to talk about clinical application here, and that is that there's an entire class of diseases called the motile ciliopathies. Now, these are genetic diseases that result from defective ciliary beating. And if those cilia in the multiciliated cells can't beat, then you can get hydrocephalus. You almost always get chronic airway infections, usually leading to a requirement for a lung transplant in the late teens and early 20s of these patients, and in many cases, infertility as well. Now, this being the GSA meeting, we don't actually study this in any of these cell types, but instead, we study multiciliated cells in a model organism. We use Xenopus embryos for this. Now, multiciliated cells in vertebrate animals were actually discovered on the epidermis of amphibian embryos, so there's a 200-year history. of this. There's some nice histology from 1896. And of course, the methods have gotten slightly better in the meantime, and we can look at the structure and also the function and the dynamics of these cells using Xenopus embryos. And so for quite a few years, we've been interested in this evolutionarily conserved transcriptional circuitry that drives the specification and differentiation of multi-ciliated cells. Now, this work really started with Chris Kentner over 20 years ago when he found that Notch and delta signaling were essential for the specification of multi-ciliated cells. And since then, his lab, our lab, several other labs listed below have established this sort of regulatory circuitry that drives the specification of these cells. But as I said, specification of the cells isn't the final picture. We want to get to the actual biological function of these cells. And so how can we bridge that gap between genomics and cell biology? And so just as an example of the gap that we need to bridge, if you take just one study, and this is a paper from Mei Zhong and Taejun Kwan when they were in the lab in 2014, performed a genomic screen of RFX2 targets, and we actually found over 900 direct transcriptional targets of RFX2. And the question was, okay, what do these 900 genes do? How do they all function? Now, sure, you can cherry pick the top couple of hundred of known ciliary genes, known planar cell polarity genes, these kinds of things. But what about all these other genes? What do they encode? What do those proteins do? And so when Fan Tu was in the laboratory, he took a protein localization screening approach. And so he actually expressed about 350 of these RFX2 targets in multiciliated cells in vivo and looked at their localization. He, of course, found a lot that were in the cilia, as you might expect, but he found a lot in a whole bunch of different organelles, pretty much every part of the cell. And what I want to focus on today is one class of proteins he called dots. I told him this is real science. We can't call them dots. We have to call them foci. And this wasn't a small class. Actually, 8% of all the proteins he saw localization patterns for, he found as foci on the cytosol. And one of the most interesting ones was an axonemal dynein. So this is a molecular motor protein that's required for ciliary beating. And this is a protein that's known to be in the axoneme. So of course, if you look in confocal microscopy, you can see that it labels the ciliary axonemes. But what Fan noticed was that it also labels cytosolic foci. And at this point, the work was picked up by Qianjie Li and Ryan Huizar, who had noted that actually several other proteins also were localized to these foci in the cytosol in multiciliated cells. And that really led us to begin wondering, what are these dots in the cytoplasm? Well, one of the most exciting discoveries in the last 10 years has really sort of been a paradigm shifting change in how we think about compartmentalization of the cytosol. And it's now known that liquid-like organelles are an important mechanism for that. And the reason that's so exciting for us, you have to understand the reason why this is so exciting to us. We have to go back and talk a little bit more about these axonemal dyneins. So axonemal dyneins, as I said, are these multi-protein motors that localize to the axonemes of cilia, and it's the microtubule sliding action of these dyneins that drives the beating of the cilia. But they're not assembled there. The multi-protein motors are actually assembled in the cytoplasm, and they're trafficked up into the axoneme by intraflagellar transport. And so these subunits are generated in the cytoplasm, and they're assembled together with a set of chaperones called dynein arm assembly factors, specific category of co-chaperones that are necessary for the assembly of these proteins. Now, it's actually really interesting that modal ciliopathy from the human genetics standpoint has been shown to result either from mutations in dynein subunits or from mutations in these assembly factors. And both of these genetic causes have their origin then in a cytoplasmic function, not an axonemal function. And so nothing really was known about the cell biology of this process. And so we thought this identification of foci in the cytosol for some of these proteins might give us an insight. And in fact, in 2018, we published a paper in eLife where we described what we now call DynAPs, or dynein assembly particles. And these are evolutionarily conserved multiciliated cell-specific organelles that contain these assembly factors, these DNAAFs, as well as the dyneins and also the chaperones. And about the same time our paper came out, Steve Brody and Amjad Hirani showed similar things in the mouse and the human airway. Pleasantine Mill has now studied these in the mice as well. Zhao Zhesun has studied these in the zebrafish. So these are really conserved across all vertebrates. And one of the interesting things about this is we've now found a large handful of proteins that localize here. This is actually a pretty small set now. It's a little bit outdated, but over half of the proteins that localize to these structures are human ciliopathy loci. So we thought then that this might be an entry point to gaining a better cell biological understanding of these human diseases. So what are these DynAPs? They're not membrane bound. So if you look at co-localization with DynAP markers and vesicles or DynAP and Golgi or really any organelle that you want to look at, you're not going to find any co-localization. Instead, they behave like these liquid-like organelles, similar to stress granules or P-bodies. Now, they're less like P-bodies, which tend to be fairly spherical, and they look a lot more like stress granules, which tend to be irregular and frequently show this fission and fusion behavior that I just showed you right there. Now, to get a better handle on the protein dynamics within these organelles, Chen-Jay Lee and Ryan Huizar performed FRAP analysis. And what this revealed is that the assembly factors display a very rapid turnover, very similar to a variety of different FRAP curves that have been identified in liquid-like organelles. So here is one of these DynAPs. You're going to see a FRAP, about half of the DynAP there, and then you're going to see the recovery here. If you just look at the still frames here, here's the pre-bleach. You bleach half of it and it recovers pretty quickly, just a few seconds. If you bleach the whole thing, again, it recovers from the cytoplasm. So this is a classic behavior of protein dynamics within these liquid-like organelles. Now, we think that these kinetics are important to their function because if we model human ciliopathy, we actually find that disrupting human ciliopathy genes actually leads to defects in the structure and the dynamics of DynAPs. So here what we've done is done a knockdown of DNAF5. This is one of those assembly factors I told you about. It's also called HEAT-R2. And what you can see is if we knock down HEAT-R2, the cilia are perfectly normal. They look a little frazzled, and that's not uncommon for cilia that don't beat. The thing I really want you to look at is here are the normal dyning arms deployed out to the ciliary axonemes in the control, and these are completely eliminated, almost completely eliminated from these HEAT-R2 knockdown axonemes. Now, if we look in these same cells and we look down on the cytoplasm, what we see is a dramatic reduction in the number of DynAPs, less than half the number of DynAPs per cell. And if we go in and we look at what happens to the kinetics of KTU or DNAF2, which is another one of these assembly factors, you can see that the assembly factor kinetics have been altered by knocking down the other assembly factor. So we think that there's some sort of complex relationship between these proteins and their kinetics in these organelles and their ability to assemble dyneins and deploy them into axones. So this led us to look a little more closely at protein kinetics in these organelles. And that's when we found something really interesting. When Chen-Jay and Ryan did FRAP analysis for the dynein subunits themselves, they found a very, very different result. So here's another one of these DynAPs. And I'm sorry, the scale bar is missing, but this is a couple of microns across. And if they FRAP this guy, half of it, it actually never recovers. And here's the FRAP, you get this tiny little bit of recovery, and it just sort of pokes along here. And in fact, if you take this out as long as you can image it, much longer timescales than we've looked at the assembly factors of the chaperones, we actually see that it just never recovers. And so if you compare then this assembly factor, and this is just a representative one, in fact, you can look at all the assembly factors or the chaperones, HSP chaperones, cofactors, et cetera, they all have this very high mobile fraction. And if you look at any of the dynein subunits, they look very, very different. So this led Chen-Jay to think a little bit harder about these dynein subunits and dive a little deeper into what might be going on there. So before I can tell you about those data, I need to tell you a little bit more about the dyneins. There's actually two general flavors of dynein arms. The outer arm dyneins are what are responsible for ciliary beating. The inner arm dyneins are actually required for tuning the ciliary waveform, and they're on different parts of the microtubule doublet within the axon. And what Chen-Jay found is very interesting. If she goes and looks then at, here's a cell right here, and here's just one little DynAP. And if you look at a chaperone and an assembly factor, you always see this very, very strong co-localization, almost perfect co-localization. However, if you look at an outer dynein arm, for example, co-localized with an assembly factor, it's actually a partial co-localization. You can see down here, it's nicely localized, but there's region here that's labeled by the assembly factor that's not labeled by the outer dynein arm. Turns out what that region is the inner dynein arm. So if you look at the outer dynein arm subunit and an inner dynein arm subunit, we actually get very, very little overlap. If, however, you look at two different inner arm or outer arm subunits, say this yellow guy over here and this green guy over here, they're going to overlap perfectly. And so what this then says is that even though this thing displays this very liquid-like behavior with chaperones and assembly factors moving back and forth in here, they actually contain very stable and what appear to be functionally partitioned subdomains within this organelle. And so Chen-Jay wanted to really begin to understand what is going on here. And one of the things that we like to do in the Wallingford lab is to think about big data sets and how we might be able to apply, say, interactome maps to problems we want to study in the lab. So interactome maps are immensely powerful tools for understanding cell biology. A couple of fairly recent efforts in this area came from Tony Hyman and Matthias Mann, from Wade Harper and Steve Gigi. And these have been very, very powerful tools, but they do have a limitation. And that is that they're really focused on a single cell type, and it's a differentiated cell type. For example, HeLa cells or 3T3 fibroblasts. We actually have very, very, very little protein interaction mapping data for embryonic tissues from any animal. So together with my friend Edward Marcotte, who's a systems biologist at UT Austin, we've put together a workflow and begun sort of the heavy lift of interactome mapping in vertebrate embryonic tissues. And the way we do this is we get a bunch of developmental biologists around, we cut 25,000 tissue explants, we grind them up, we give the protein or the RNA to systems biologists, they assure me that hypergenetic probability actually is a thing, and then they give us back Excel spreadsheets, and then we go and do cell biology. And this is actually working pretty well so far, and I'm just going to take you in the last part of my talk through a few of these results. So first thing we did was to look for interaction partners with the outer dining arms and the inner dining arms. And if Chanjay pulls down an outer dining arm subunit here, DNAi2, the yellow one here, she finds all these other outer dining arm subunits like she's supposed to. But she also identifies some novel proteins here, and the most interesting one is this totally unannotated and totally unstudied protein called C16ORF71. Both in Xenopus and human, this is data from our collaborator Amjad Hirani at WashU. C16ORF71 is actually a DynAP-specific protein. It's not present in the axonemes. So here's the axonemal dynein. You can see it labeling all the axonemes. You can see it labeling the DynAPs here. This is a DynAP-specific protein. In fact, when Chanjay looks at Xenopus, she can show that it's actually specifically in the outer subregion of the DynAP. And sure enough, when she knocks it down in Xenopus, she sees a complete loss of outer dining arms and essentially normal inner dining arm deployment. And so it looks like C16ORF71 is a DynAP-specific protein required specifically for the ODA deployment to axonemes. Now, if she pulls down an inner dining arm, in this case WDR78, she gets a different set of interactors. So she gets a lot of the things that she should get, these other subunits of the axonemal dynein. She also found WDR18, which is an unstudied protein but implicated in cilia feeding. She also gets the splicing factor, SF3A3. Now, we thought this had to be a red herring. It couldn't possibly be right. But sure enough, when we made a GFP fusion to this protein, it localized very nicely to DynAPs. Now, that's not surprising because a lot of liquid-like organelles actually contain RNA. So Ryan Weizar went and looked at whether or not DynAPs contain RNA, and sure enough, they do. Here's just a soluble RNA marker or cell-permeable RNA marker. You can see it lighting up the nucleus here, and you can see it lighting up these DynAPs labeled with DNAF2. And this paper is actually up on bioRxiv this week, and I encourage you to take a look at that. So this idea that there are RNAs in there wasn't terribly surprising, but it did then lead to the question, what are those RNA-associated proteins? And for this, we went to another interactome mapping approach that was developed in Edward Marcotte's lab by Anna Mallon and our joint postdoc, Kevin Drew. And in this approach, what we do is we're going to isolate tissues, and this is just a Xenopus animal cap, which actually differentiates into mucociliary epithelium. We isolate the proteins, we can split the sample, we can treat one of those aliquots with RNase, we leave the other aliquot alone. Then we can perform size exclusion chromatography. And what's going to happen is that an RNA-associated protein might shift its elution pattern in the presence of RNase, and we can detect that by doing mass spec on all of the fractions and look for these elution shifts. And this is actually a paper last year in Cell Reports where they applied it to a variety of mammalian cell types. We've now applied it to Xenopus embryonic tissues, and it's actually working quite nicely. So here's just an example of what the data look like. This is a ribosomal protein. You can see a nice shift in the presence of RNase. This is another one called nucleolin, a nice shift in the presence of RNase. There's a nice computational framework where we can put a confidence score to this. Here are these guys up here that we expect to see shifting. And then what's exciting is we can go look in there for proteins that we thought might be related to motile ciliogenesis or motile cilia function. And sure enough, one of our very high scoring proteins is this Chlamydomonas flagellar associated protein 44. Now this is exciting because CFAP44 is actually known to tether the inner dining arms to the axoneme. But why is it an RNA binding protein? There's really no reason that it should be binding RNA in the axoneme. And so we went looking. And what Chanjay found is in Xenopus, CFAP44, just like Chlamydomonas, is nicely enriched in the axonemes, right where we expected it to be. But what wasn't known and that she discovered and is very exciting is CFAP44 is also localized to DynAPs. Not only to DynAPs, but actually to the inner sub-DynAP, exactly where the inner dynein arms are. And so what we think then is that this interaction with RNA may be related to maintaining this protein or recruiting it to DynAPs so that it can then assemble together with that inner dynein arm before it's deployed to the axoneme. And finally, a rotation student in the lab, V. Dang, actually now modeled CFAP44 based on the RNA binding protein Gemin-5, and we can get a homology model for this protein. So we're actually very excited now to go in and actually test some point mutations that would impact the RNA binding of CFAP44 and ask if we lose this localization. So what I've told you here then is about a new liquid-like organelle, and this is pretty exciting because liquid-like organelles provide a mechanism for diversification of cellular functions during development. So there's a whole constellation of different liquid-like organelles that are very well known. Some of these are cell type specific. The oocytes have germ granules and bulby antibodies. I've told you today that multi-ciliated cells specifically have a new organelle called DynAPs. I told you that these are novel. They're specific to the multiciliated cells, and the evidence suggests that they're sites of axonemal dynein assembly. But like so many organelles that are liquid-like or phase separated, it's been hard to really determine whether the phase separation and the concentration of these DynAPs is an essential part of their function. We're working on that now. What I can tell you though is that they're functionally partitioned for outer dynein arms and for inner dynein arms. Showed you that C16-ORF71 acts in DynAPs to regulate outer dynein arm assembly. I showed you about a new method called DIFRAC that's identified some RNA-associated proteins that are present in the DynAPs. And finally, once again, I want to circle back around and thank my lab. I'm blessed to get to work with such a great group of people. This project was really mostly spearheaded by my senior scientist who's been with me for a long, long time, Chanjay Lee, and actually her stellar undergraduate, Ryan Weezar. He's now ending his first year as an MD PhD at Johns Hopkins University. The in vivo screen was done by Fan2. We had a tremendous amount of help with the DIFRAC work from Kevin Drew. And for all the proteomics, we absolutely couldn't have done it without Edward Marcotte's lab. Ophelia Popoulos and Rachel Cox were hugely involved. Also Claire McWhite and Caitlin Devitt worked quite a bit. And our collaborators at Washington University for the human and mouse work are Amjad Harani and Stephen Brody. I'd like to thank these people for funding. And finally, because I can't take any questions, what I've been doing with the pandemic is learning how to navigate social media. I've opened a Twitter account, much to the derisive laughter of my lab members. And if you want to tweet me your questions at JB Wallingford, you're welcome to do so. Thank you very much for listening.
Okay, John, thank you. Incredible micrographs that you were showing. And I am going to ask you one question that came up, and that is, what are the functions of cilia or multi-ciliated cells on the epidermis?
Yeah, that's a great question. So it's still not entirely known, but it's thought to be twofold. One is innate immunity, just like the airway. By keeping fluid flow going across it, you have fewer little microbes and bugs that can stick to the epidermis and infect it. But another major function is thought to be respiration, because actually the cilia disappear from these cells. There's a great paper just out in bioRxiv from Peter Valentek's lab studying this multiciliated cell disappearance, and that seems to correlate with the onset of gill function. So I think respiration is probably the major function of the multiciliated cells in these organisms.
Okay, great. All right, I want to thank Elaine and John, and I want to congratulate both Jerry and David for well-deserved awards. And this brings this session to an end, but I have one thing I want to say, and that is we have a lot of people attending, and one of the best ways to support GSA and all of the great work that we're doing is go to the website and become a member. And while you're there, clicking the member site, next to it is a donate site, and throw $50 into the bucket, and that will help us keep producing meetings like this and all the work that the GSA staff does. So thank you, everybody.